All right, let's move on. There's your learning objectives. Use them as you see fit. This is another relatively dense lecture, so I got to keep up the pace. Anyway, right now we're going to talk about translation. So we already talked about DNA replication, talked about transcription. Now we're going to talk about translation. And what is translation? That's actually the protein synthesis. So those two terms are used interchangeably. So, and for some reason, some people get that confused sometimes. Protein synthesis is translation and vice versa. So the code in that messenger RNA we made in the last lecture is going to be translated into a protein. So the mRNA and also the transfer RNA and the ribosomal RNAs, they have a role in translation as well, as we're going to see, are going to be used to complete translation. So all three of the major classes of RNAs. So the protein coding region of a messenger RNA, now you remember there's a coding region and there's an untranslated region on each end of a messenger RNA, right? We'll talk a little bit about those later, but the protein coding region contains a series of 3 nucleotides. So they're grouped into triplets. And each triplet is a codon. Now, you know that in RNA you can have one of 4 different nucleotides at each particular spot. So if you do the math and you have a triplet, then you can come up with 64 possible combinations of 3 nucleotides. Okay? So the order is important as well. And when we look at our genetic code, interestingly enough, we're going to have all 64 possibilities recognized there. The nucleotides are actually read 5' to 3'. So during translation, we're going to move along the messenger RNA 5' to 3'. And the protein is synthesized from its N terminus to its C terminus. So the N terminus is the beginning of a protein. And it actually contains a free amino group. That's why they call it the N terminus. And the very last amino acid added contains a carboxylic acid group, a free carboxylic acid group. That's why it's called the C terminus. Okay, so one of the things you need to be able to do is use this genetic code table to figure out what a codon actually encodes. Okay, and so we're going to walk through one real quick. This is something that I've found that students have already experienced, so they don't need a lot of help on. If you struggle with this, come see me in my office. We'll get you straight in about 2 minutes. But I don't want to take too much of the class time talking about this. But we'll just look at AUG to begin with. So how do you use this table? Okay, we have AUG codon. What does that encode? How do we use this table to figure that out? Okay, well, the first thing you do is you start over here on the left, and the first nucleotide is A, so you've now defined it as this row. The next nucleotide, we go to the top. It's now defined as this column, so now we've narrowed it down to these 4 nucleotides. And then the 4th nucleotide, in this case, could be U, C, A, or G. In this case, it happens to be G, so it's right here, methionine. So that's how you do it. Boom, boom, boom. Okay? I think most people don't have too much of a hard time with that, so I don't want to spend a lot of time on it. Now, in this case, this actually happens to be the start codon as well. So translation always begins with an AUG codon. The AUG encodes methionine. Methionine actually only has 1 codon. So if there's other methionines in the protein, they're going to also have an AUG codon. So not every AUG codon is a start codon. So only the very first one is considered the start codon. And I'll explain how we define the first one for eukaryotes and prokaryotes a little later in this lecture. So one of the things I'm not going to ask you to do is try to memorize the genetic code. I don't have the genetic code memorized, okay? I do know what the start codon is, and I do know what the stop codons are, and I would ask you to know those. So the start codon, there is only one. AUG happens to encode methionine, okay? And so on a board exam, it's fair game to know what the stop codons are too. So I've given you some little mnemonics there to help you try to remember. UGA, UAA, and UAG are the 3 stop codons, as you can see in here in this genetic code. UGA, you go away, UAA, you are away, UAG, you are gone. That's kind of how I remember what they are. And I learned that 15 years ago, and I still remember those today. So if you get those straight in your head, you'll remember the stop codons. So when would this ever be used? Well, I could give you a sequence where you needed to know what the start codon is, because that's where you begin translation. You need to know where it stopped. And even without the genetic code, you could tell me how many amino acids were there. And we'll do a little exercise like that shortly. The first stop codon is defined, as defined by the open reading frame, is the stop codon. So as you're going along, you hit a stop codon, you're going to stop. Doesn't matter if there's 100 more downstream, you've already stopped. Okay, so it's an orderly event in this process of translation. So a little bit about the genetic code. Some terms. Firstly, the genetic code is specific. That means each codon is specific to a certain amino acid. Any codon I give you, and I give you the genetic code, you can actually look it up and you will find a very specific amino acid, or perhaps a stop codon. It's one of those 3, but it's very specific. It's universal, so the genetic code is the same for all organisms. Now there are a couple exceptions there. Your mitochondria in different organisms are a little bit weird and have a little bit of a different genetic code, but actually even there, most of the genetic code is actually identical. There's just a couple differences. That means I can take a gene from humans and put it in E. coli and translate it. Okay? It may not undergo all the correct post-translational modifications, but it will certainly be translated in terms of the primary sequence. Okay, the genetic code is also redundant, so we have to contrast that with specific, because that sounds like they're contradicting each other. Redundant means that each amino acid may have more than 1 codon. Some of them only have 1 codon. Methionine was an example there. But some of them have multiple codons, even up to 6 possible codons. Okay? So if you're starting with the amino acid, it's redundant because there could be multiple codons. But if you're starting with the codon, it's specific because it can only encode 1 particular amino acid. So that's what we mean, depending on which direction we go in. So don't confuse those 2 terms. The genetic code is also non-overlapping and commonless. So it's a continuous series of 3 bases. Once we define the start codon, the next triplet, the next triplet, and the next triplet are all defined. There's no overlap. Overlap would mean that you used 1 nucleotide twice. No gaps in there where you skipped a nucleotide. No codon is read twice. Okay, so once you define the start codon, you've defined everything downstream. Okay, now those of you who've studied virology may know that there are some exceptions there, but we're not going to talk about them right now. So here's an example of an exercise I could give you. How many amino acids does the protein encoded by this messenger RNA contain? Okay, in this case, we can tell this is a eukaryotic messenger RNA because we have a cap and a 3' poly A tail. And the way you determine this, first of all, you need to realize the untranslated regions aren't grouped in triplets. You only group things in triplets once you define the start codon. So the untranslated, and that's one of the things that always screws people up with these types of exercises. You don't go three nucleotides, you don't define anything until you find that start codon. And in eukaryotes, we actually start by starting at the very 5' end and moving along until you find the very first AUG. And there's other AUGs in there, okay? There's an AUG there, depending on what the reading frame is. But you go along until you find the very first one, okay? And there is the very first one. Now we define the reading frame. Underline that. Every 3 nucleotides beyond that, we can divide it up from there. And you just go along till you find a stop codon. Okay, we divided it up into 3. Not a stop codon, not a stop, not a stop, not a stop, not a stop. Oh, there's a stop. Okay, that's where it ended. So it doesn't matter what's going on after that. How many amino acids are there? Stop codon is not an amino acid, doesn't encode an amino acid, so that doesn't have an amino acid. So we got 1, 2, 3, 4, 5, 6. AUG, the methionine, the start codon does encode an amino acid, so the answer there is 6. So that's an exercise I could ask you to do. Now, if I give you the genetic code, I could even ask you, why don't you tell me what the sequence of that polypeptide is? And you could use those codons and actually read this table, and I've actually went through and did it for you. But make sure, try it on your own, and make sure you get that particular answer. Okay. Once again, it'd take a long time for me to walk you through all that right now. I think most people don't struggle with that too hard, so you can walk yourself through that. Once again, if you have problems, we can come in my office. We'll draw some pictures, and we'll work out some problems, and you'll have it straight. Okay, so one of the things that we talk about in the section on translation is mutation. So if we have a DNA sequence that gets mutated, we have several outcomes possible, multiple outcomes possible. First, we're going to look at our single nucleotide changes or our point mutations. Okay, single nucleotide change is a point mutation. We can have a silent mutation. That means we've actually changed the DNA sequence, but when we look up on the genetic code, we ended up encoding the same amino acid. Okay, and they've given you an example here. There's the serine codon. We've changed one of the nucleotides, but when you look it up in the genetic code, it's still a serine. Okay, that's a silent mutation. Shouldn't affect the function of that protein because it's going to have the same amino acid. Missense mutation, that's where we've changed a nucleotide and now we encode a different amino acid. Okay. So in this case, they're showing you an example where they change one nucleotide. If you use your genetic code or the genetic table and look it up, it went from a serine to a proline. That's a pretty big difference there. Now, that proline has a weird structure, so that could potentially cause problems, but it's certainly an example of a missense mutation. You could also have a nonsense mutation. Really, the missense and the nonsense are the two that people sometimes mess up. So the nonsense mutation is where you go from something that encodes an amino acid to something that is now a stop codon or a termination codon. Okay, and they've given you an example. They've mutated one nucleotide, and now we went from a serine to a stop codon. That is a nonsense mutation. So the way that I remember the difference between nonsense and missense, which are all the others are real pretty straightforward. The name kind of tells you what's going on there. But missense and nonsense are the ones that sometimes throw people off. So I just remember non-stop, so nonsense. So molecular biology is nonstop fun, right? I'm sure this week's been a blast so far. Just can't wait to get to some more, or whatever else you want to say is nonstop. But nonstop, that nonstop helps me. That's how I remember nonsense is a stop codon mutation. Okay. You can also have some other types of weird mutations. So those last examples were point mutations. You can also have trinucleotide repeat expansions. And there's some very big, high-yield board exam diseases which are trinucleotide repeat expansions. We're not going to have time to get into them today, but they're listed there. 3 of the absolute biggest ones, and some of them you've probably heard of: Huntington, Fragile X, myotonic dystrophy. We'll certainly talk about those later on in genetics. They're all examples of trinucleotide repeat expansions. So these are regions where there's already in the normal gene a trinucleotide repeat. Okay, that means 3 nucleotides are repeated over and over and over again. So even in the normal gene, that happens. But what happens is somehow during DNA replication, these nucleotide repeats confuse the polymerase, and it actually amplifies it. And once it amplifies it and it gets bigger, that's when it gets to a certain point can actually cause a disease. 3 huge major diseases we're going to talk about are listed right there. But that's what it is. We've just expanded that trinucleotide repeat. If it happens to be in the coding region, we're actually going to make a bigger protein with the same codon repeated over and over and over again. But it can also, weirdly enough, happen in the non-translated region. And I'll explain when we get to those diseases how that actually results in disease. You can also have insertions or deletions. In this case, so we have to differentiate. There's a couple different ways you can get insertions or deletions. So if it's not a multiple of 3, we're going to have a frame shift mutation, right? The first start codon defines the triplets. If you add or remove some nucleotides in there, and let's say you added 1 or added 2, well, now that's not a multiple of 3. It's actually going to redefine everything from that point now as a different reading frame. And what you'll actually find out if you do that, and you can do it yourself for fun if you're really bored, insert a nucleotide in there, everything downstream when you translate it now becomes a completely different protein. And very often when you do that, it also introduces an early stop codon, and you end up with a completely different protein that's also truncated, so it's not the same size. Now, if you have an insertion or deletion that is a multiple of 3, because the nucleotides are grouped, the codons are grouped as triplets, what that ends up with is if you add 3 nucleotides, now you just had an extra codon in there. So you don't disrupt the reading frame, so you have the same, the C terminus of the protein ends up being the same sequence. So whether you insert or delete 3, you'll just insert or remove a codon, and 6, and 9, and so on. You can also have splice site mutations. We talked about splicing in the last lecture. I told you you didn't have to memorize what the splice site sequence was, but it is specific. So if you change those nucleotides, and in some cases they're nucleotides that could be in the intron, for example, so they shouldn't even end up in the express sequence, but it screws up the splicing, you'll end up splicing incorrectly, therefore you get an incorrect protein, in some cases an unstable RNA. But anyway, you don't make the correct protein, and there is some major diseases that are caused by splice site mutations we're going to talk about. Okay, so next we're going to look at the components that are required for translation. We're going to go through all the components and then look at translation itself. First of all, obviously, you have to have the amino acids. Typically, proteins are going to have all of the amino acids in there at one point or another. Proteins, on average, tend to be hundreds of amino acids long. Certainly, they can be even bigger than that. They can be thousands long. There are a few that are less than 100, but your average one is going to be hundreds of amino acids long. So usually, all the amino acids are in there at some point. If you do have an amino acid that isn't particularly limited, what ends up happening is the ribosomes end up stalling at that point, and then they can't continue translation. You have to have at least 1 transfer RNA for each of the amino acids, and in many cases you have more than 1 transfer RNA. You don't have 64, you don't have 61, so there's actually fewer, and we're going to explain why that is a little bit later. Amino acids that are specified by multiple codons often have multiple transfer RNAs. There's on the order of 35 transfer RNAs, and I'll explain why there's fewer than you might predict. Transfer RNA is going to have the CCA terminus. Okay, we talked about that's added post-transcriptionally. That's where we're actually going to attach the amino acid. Okay, if the amino acid is already attached, we call that a charged transfer RNA. If the amino acid is not attached, it's an uncharged transfer RNA. Okay, so we have to charge the transfer RNAs in order for them to be used in translation. And the transfer RNA also contains the sequence called the anticodon, contained there in the anticodon loop, that's actually going to base pair with the messenger RNA. Okay, just the same base pair rules we learned before. So that's how we know that this transfer RNA, or that's how it matches up correctly with the particular amino acid. Okay, so AUG, you remember that's methionine, right? There's methionine. And actually you can determine what the anticodon would actually be, just using your base pairing rules. We have these enzymes, aminoacyl tRNA synthetases. These are actually true molecular dictionaries. These are really cool enzymes because they actually recognize a nucleic acid and they actually recognize an amino acid. So they recognize two very different things and are able to join them together. So you have one aminoacyl tRNA synthetase for each of your 20 common amino acids, and it will recognize all of the transfer RNAs for that particular amino acid. And what its job is to actually attach the amino acid to the correct transfer RNA, attach it to that CCA site. You end up cleaving two high energy bonds, so this is just like when we added a nucleotide to DNA or an RNA chain when we're making it. We actually are going to use energy. We're going to cleave that pyrophosphate to inorganic phosphate, drive that reaction forward. I told you we're going to see that over and over and over again. Okay. These tRNA synthetases actually do have proofreading and editing activities. That means when they add an amino acid to a transfer RNA, they're going to check and make sure that it's correct. And if it's not, they're going to cleave it and then start over again and try to make a correct version. So they do have proofreading activity. A little different than the proofreading activity we talked about for DNA polymerase. Obviously, we need a messenger RNA that's going to be used as a template to be translated, and we need our ribosomes. Ribosomes, as we mentioned before, a complex of RNA. There's 4 in prokaryotes, 3 in prokaryotes, 4 in eukaryotes, and then many proteins. I don't even get it. It does say there, 50 proteins in a prokaryotic ribosome, somewhere on that order. It's a lot of different proteins. Very complex machine. I already asked you to learn the sizes of those, so that's just to remind you once again. It's important. You'll just have to trust me right now to know those later on. The subunits, so ribosomes have 2 subunits, and they actually exist separately in the cytoplasm until translation starts. Then they come together. There's also different spots on the ribosome and the AP and the E site. And I'm going to get to those again. So I'm going to wait till I have the figure where it shows the AP and the E site, and I'll show you what those do. A site's where the incoming amino acid is. The P site's where the peptide that's been formed so far sits. And then the E site's where the last transfer RNA that's got cleaved now is going to exit. So the only other thing I had here was the endoplasmic reticulum. A lot of the translation actually occurs on the endoplasmic reticulum, which we're going to expand upon a little bit tomorrow. And so there's a rough endoplasmic reticulum, and that rough appearance is actually those ribosomes on that particular version of the endoplasmic reticulum. And proteins translated on there are, as we're going to see tomorrow, designated for particular areas. There's multiple different protein factors in translation. Those of you who've taken a class where you got into some real fine details on translation may have seen some of these. Initiation factors, elongation factors, termination factors. We're only going to look at one specific one because it's related to a disease. The rest of them, we're just going to call them initiation, elongation, and termination factors. There's a lot of them, and you're free to go memorize them if you want, but they'll never be asked on a board exam, so we're not going to spend time trying to memorize those. It requires translation, just like making RNA or DNA requires a lot of energy. Charging the transfer RNA itself is where some of that energy came from. Okay, so 2 high energy bonds were used, but binding the incoming amino-acyl transfer RNAs we're going to see, that requires GTP, translocation. So that's when the ribosome moves to the next triplet. Actually requires GTP as well. So every amino acid that you're going to encode requires 4 high-energy bonds that get cleaved. And here's our first example, at least the first example I've talked about, where we're actually using GTV as an energy source as well. One of the things I would ask you to be able to do is determine an anticodon if I give you a codon. You just use your same base pairing rules, okay? And I've given you an example right there. Or I could even give you, I could give you an anticodon. To give you an anticodon and say, what amino acid does that encode? Well, in order to do that, what you have to do is you have to determine the codon first. You don't look up the genetic code tells you codons, not anticodons, right? So you have to determine the codon first, then look it up. It's an example of something that we could do. Once again, this follows the same rules, so I don't think it's terribly difficult. If you're struggling with it, come and see me. I've given you an example there. You can walk through that example yourself in the interest of time. I'm not going to walk through it because it'll take a few minutes. But you can try it yourself. If you struggle with it, come see me. Okay, so the next thing is the wobble hypothesis. Now, I mentioned earlier there isn't 61 different transfer RNAs. Why did I say 61 versus 64? We have those 3 stop codons. There isn't a transfer RNA for a stop codon. We have termination factors that recognize stop codons. So you predict there's going to be 61 transfer RNAs, and there's not. There's somewhere on the order of 35, 40, somewhere in there. It really depends on the organism. But anyway, there's not 61. So what's going on there? So transfer RNAs can recognize more than one codon. How in the heck can that happen? Now, first of all, I want to mention real briefly, if I ask you to do something like this, just assume exact base pairing. Forget the wobble hypothesis when we're talking about this. I want you to understand what the wobble hypothesis does tell us. So you can have a transfer RNA that recognizes more than one codon. That means, okay, if it's got a very specific sequence, then the sequences can't line up perfectly. You can't have that perfect base pairing that we've seen before. So what happens in a transfer RNA or in a messenger RNA, the third nucleotide in a messenger RNA, which happens to be the first nucleotide, because remember we have anti-parallel, on the anticodon, those can sometimes bind in non-traditional ways. The first 2 nucleotides on the messenger RNA or the second and third on the anticodon are always an exact match. But sometimes that third one is not exact. Okay, and there is some rules. It can't just be anything. There is some rules, but I'm not going to ask you to learn the rules, like what are the unusual matches that can occur. Don't try to memorize those. Just recognize there are some that can occur. Okay? And so what you actually end up finding, and you can go back and look at the genetic code yourself, you'll find that very often the third nucleotide in a codon doesn't really matter. Okay, like you'll find the third nucleotide, no matter what it is, all encodes the same amino acid. That's not always the case, but in many cases it is. So that's kind of how one of the neat things you can see on the genetic code table. Okay, now we're actually going to look at the process of translation. We've defined genetic code, mutations, all those things. We've defined all the things we need to do translation. So now we're actually going to look at translation, going from the 5' end to the 3' end of the messenger RNA. So most of this is just a repeat, just a review. One of the things we're going to explain as we go through here is why eukaryotes are always monocistronic, has to do with the process of translation, and why prokaryotes can be polycistronic. Okay, so all this stuff I've actually already said at some point or another. So what we're going to do is we're going to go through a model for prokaryotes, and where the eukaryotes differ, we're going to explain that. Okay, and actually one of the places they differ is at the very beginning, which is in the initiation. Okay, in prokaryotes, we initiate translation, It's potentiated by the presence of a Shine-Dalgarno sequence. So I do want you to know what a Shine-Dalgarno sequence is. Okay? It's used in prokaryotic translation. It tells the ribosome where to begin translation. Don't confuse that with promoter sequences, which don't end up in the messenger RNA. That's a typical mistake I see lots of students make. Shine-Dalgarno sequence is not a promoter sequence. It's a sequence that's present in the messenger RNA that tells the ribosome where to bind. It lines it up correctly with the first start codon. Okay? So you actually have a complementary base pairing in one of the ribosomal RNAs with this Shine-Dalgarno sequence that allows that small ribosomal subunit to be lined up correctly with the beginning start codon. Okay. In eukaryotes, that's different. So so because we have this Shine-Dalgarno sequence, we could actually put a Shine-Dalgarno sequence anywhere on the messenger RNA, right? Put it in the middle, we could put it at the end, and then have a start codon. So we can actually make a messenger RNA that has multiple different reading frames or encode multiple different genes. In eukaryotes, we don't begin translation this way. In eukaryotes, what happens is a small ribosomal subunit binds to the very end where that cap structure is, that 5' end where the cap structure is. And what it does is it moves along till it finds the very first AUG, which is similar to that exercise we did at the beginning of the lecture. Okay, so it scans. It's called the ribosomal scanning. Moves along until it finds the very first AUG. That very first AUG defines the open reading frame. Translation happens till it hits a stop codon, then it falls off. Okay? So that means it doesn't matter if there is other start codons down there, or if you make a synthetic one and you stick it in a cell and see what happens, it'll still always only translate that very first one because it has to start from the 5' end. Once it hits the very first AUG, that is the start codon, and it now has defined the open reading frame for that messenger RNA. Okay? So that's why you can't have polycistronic messenger RNAs in eukaryotes. You can make them in the lab and stick them in, you'll still only get one protein produced from the messenger RNA. Okay. Okay, so the initiator transfer RNA, so now we've got it lined, we've either got it lined up correctly or we've scanned along till we found the first AUG, depending on whether we're talking about prokaryotes or eukaryotes. And this model in this picture is actually showing you a prokaryotic model. Next, we have the initiator transfer RNA, which is going to recognize this AUG start codon. Okay. In eukaryotes, you have a special one, and it happens to be a formulated methionine. The initiator transfer RNA is a little bit different than in eukaryotes. In eukaryotes, the initiator transfer RNA is just bound to methionine, just like a regular methionine transfer RNA. Okay, so at this point, we just have the small ribosomal subunit. The correct transfer RNA is now there. It happens to actually be in the P site, ultimately in the ribosome. So we're going to have the large ribosomal subunit now joined. You've got to have them joined together in order to actually translate the molecule. You'll notice we're in the P spot at this point. So here's our A, P, and E spots on the ribosomes. Okay, that's where the incoming transfer RNAs are going to come in. The P site's where the peptide is going to be that we've synthesized so far, and then the E site's where the exit site is, where the transfer RNA, once it's cleaved, is going to be. So now we can actually begin translation. So the next codon there is UUU, happens to encode phenylalanine. That transfer RNA will come in, bind through the complementary sequence. It'll line up those two amino acids next to each other. Okay. And so there's a enzymatic activity now associated with the large ribosomal subunit. I think I have that written on the next slide, yeah. Which is the peptidyl-transferase, which is actually going to join those two amino acids together now. Okay, so that's what's going on here. Here's our A site, here's our P site, so the P is what was already there, and it's actually going to join those two together. Now at the very beginning, we just have the 2 amino acids, so you're just making 2 amino acids, but eventually this peptide is going to get longer and longer and longer. This elongation event itself actually requires GTP. I didn't mention here in the beginning, it's written in here, when you actually initiate translation, you require an additional GTP. I'm not too worried about that. I just want you to know for every amino acid, we're going to add 4 high energy bond equivalents. Okay, so now we actually need to move this ribosome to, so now we've made it this dipeptide, right? We have our formulated methionine, phenylalanine, so that peptidyl transferase actually joined those two together. Okay, so now the peptide has been moved here. Now the ribosome has to translocate, has to move, so that the next codon to be translated now occupies the A site, and that peptide gets moved to the P site, and then we have that empty transfer RNA in the E site. So the next time that moves, that transfer RNA, the empty transfer RNA, will be booted off. Okay, so now we have another codon there. In this case, I believe it happens to be lysine. Lysine's going to come in. We're going to use peptidyl transferase, move that over to the lysine molecule. Ribosome's going to move again. Okay, and so on until eventually, at some point, a termination codon is going to end up in the A site. There isn't a transfer RNA that recognizes that. It's going to be a release factor. So there are multiple, and we didn't talk about the specific ones. So beyond what I'm talking about here, in case you ever see these, there are multiple initiation factors also involved in this, multiple elongation factors, and there are multiple possible termination factors involved in here. These are very, very big active areas of research. And some of you may, if you've taken a class where you've talked about it in more detail, looked at some of the specifics there. Well, I'm not going to ask you to learn. There's one I'm going to ask you to learn, which we'll get to shortly, but there's some other details in there that you don't have to learn. In prokaryotes, you actually have 3 possible termination factors. In eukaryotes, you happen to know there's only 1 termination factor. And what happens is that termination codon is in that P site. So what's going to happen is, in order for termination to occur, we're going to have to dissociate the ribosomes, and you still have a transfer RNA attached to that polypeptide you made, and so you have to finally cleave that off. Okay? Messenger RNAs are normally much longer than the ribosomes. Okay, so ribosome actually occupies somewhere in the order of 50 nucleotides. Depends on what state it's in, but it's about that size. Okay, when I say occupies, I mean that's sort of the footprint it's occupying at the time on a messenger RNA. Messenger RNAs are typically thousands of nucleotides long. Okay, could be a little bit shorter, hundreds anyway. Okay, so they're much, much bigger than the space occupied by a ribosome. So what that means is once a ribosome has moved far enough along the messenger RNA, another one can come in and start translation. Okay, so you don't have to wait for the translation, a protein, to be completely translated before another ribosome can come in. So when you look at actually under the microscope, under an electron microscope, you can actually see messenger RNAs with a whole bunch of ribosomes bound, okay, in various stages of translation. So this allows us to produce a lot of protein from a single messenger RNA very, very quickly. And so here's the figures from your book. Once again, and I put them as one slide so you can kind of walk yourself through those steps. Don't go in any more detail than what I mentioned and what I have written there. If there is a little bit more detail there, I like the picture because it's nice and colorful, so it makes it fun, relatively fun to study. But don't worry about the specific factors other than the one that I'm going to mention shortly. Okay, so I already talked to you about some antibiotics. Which ones did we already mention? We mentioned quinolone antibiotics, right? Which is a DNA replication inhibitor. It inhibits DNA gyrase, so it inhibits replication. Also talked to you about rifampin, rifamycin, right? That was a transcriptional inhibitor and inhibited prokaryotic RNA polymerase. There's also a bunch of translational inhibitors that are important classes of antibiotics. These are some of the major classes of antibiotics you're going to see. You're going to learn them in much more detail than what I'm talking about right now, but I want to start to get you familiar with these. So actually, we've already looked at rifamycin, we've already looked at quinolone antibiotics. Now we're talking about our protein synthesis inhibitors. When you look at all the antibiotics in total, at least the conventional ones, they're all represented in that picture. So you've got a good introduction to a lot of them already. A couple more we're going to see in biochemistry too. So there's going to be a lot more specific ones you're going to learn, but they can be classified in one of these categories. And so I do want you to know these now. What I want you to know at this point is the ones I'm talking about right now are translational inhibitors. Okay, I'm not going to get into the specifics of exactly where they work. You will get into that in other classes later on. So, streptomycin, that's a prototypical aminoglycoside. So aminoglycoside is the class of antibiotics. It is your prototypical aminoglycoside, streptomycin. Erythromycin is your prototypical macrolide antibiotic. There's a whole bunch of others, but erythromycin is the prototypical one. So that's the most famous one and the one you'll be introduced to first. I don't know why there's an R there. It should say tetracycline, not tetracycline. Tetracycline, another class of antibiotics, the tetracyclines. There is also an antibiotic called tetracycline. And chloramphenicol, that is an antibiotic. It's also a class of antibiotics. These are translational inhibitors. Some of these antibiotics, in particular, chloramphenicol is notorious for this. One of the things you got to keep in mind, and I already had somebody come and ask me a question about this, mitochondria very much look like prokaryotes. So what ends up happening is some antibiotics can actually affect mitochondrial enzymes. Sometimes they're different enough where they don't really affect it. The mitochondrial enzymes are a little bit different. So some of the antibiotics affect it more or less. But some of them are particularly effective against mitochondria. Chloramphenicol is one of those. So it can interfere with protein synthesis in mitochondria. And some of the other antibiotics you're going to see can interfere with processes in the mitochondria. So how do these work and not kill human cells? Well, one thing is mitochondria actually have 2 additional membranes you have to cross before you can access the matrix of the mitochondria. And one of those membranes is very, very impenetrable, so it's hard to get across. So in order to get a therapeutic or a dosage big enough to affect into a mitochondria, you have to have a very large amount. So that's why dosage of antibiotics is important. You'd think if it's an antibiotic and it doesn't hurt human cells, well, who gives a crap? Let's just dose them with as much as we can, right? And some antibiotics you can do that to a certain extent. But you do have to be careful because if, like with chloramphenicol, for example, if you give too much, it will end up penetrating that mitochondria, will inhibit mitochondrial translation, and therefore could cause health problems. Okay, so this was the one elongation factor that I did want you to learn about. So just for your own knowledge, when you see elongation factors, when you see initiation factors, you'll just see the abbreviations are pretty logical. EF, elongation factor, or IF is initiation factor. When you see this little E in front of it, this lowercase e, that's a eukaryotic version. So the prokaryotic versions just have the regular names. The eukaryotic versions have a little lowercase e in front of the elongation factors and the initiation factors to indicate we're talking about eukaryotic versions. So there's a couple of bacteria, diphtheria and Pseudomonas, the toxins of those two bacteria, and actually if you look at them molecularly, they're basically the same thing. They actually work by inactivating eukaryotic elongation factor 2. Okay, so that's the one elongation factor that I'm going to ask you to know. What happens if you inhibit eukaryotic elongation factor 2? Well, you inhibit elongation. Therefore, you inhibit translation. So these are translational inhibitors that act on eukaryotes. Now, you should. One of the things I think I asked in your objectives is, and some of what you might be asking when you're going through, when you look at, okay, so we have a couple toxins here, they're affecting translation. Antibiotics, we talked about some antibiotics. They're also affecting translation. So why do we call one thing an antibiotic and one thing a toxin? Well, really, that's based on what they affect. So antibiotics are against bacteria. That's why we call them antibiotics. But they really are toxins. They're just toxins for bacteria that don't happen to really affect humans to any significant extent. We call it a toxin when it actually affects a person. So from the bacteria's perspective, they would consider this an antibiotic against humans, if they were trying to kill you. So I think we got through all of that. I know there was a few questions that I had at the end of the last lecture that I didn't have a chance. We ran out of time, so we've got a couple extra minutes. I know that was really fast. I was afraid I wasn't going to get through everything, and I ended up actually finishing a couple minutes early. So that was kind of a whirlwind. Translation, I would normally expend more than one lecture on, but only had one today. Yes, question?
For the very last slide, is that a typo of translocation instead of translation, or do you mean translation or translocation?
It's not. No, it's not a typo. This factor, this elongation factor, actually affects the translocation. So you could say it does prevent translation from happening, but it also is preventing translocation. What it actually ends up doing, that specific toxin, prevents the ribosome from moving. So it wasn't a typo, but you're correct in your thinking. It does actually affect translation. Yes? I guess I'm not sure what your question is. Are you asking about the 3' and the 5' untranslated regions interacting, or are you talking about? Yeah, exactly. So you're talking about this figure right here. Basically, what's going on here is you have there's multiple ribosomes bound to one messenger RNA, and they're just at various processes. Each one of these eventually is going to move to the end and make an individual protein subunit. They're not joined. If you did a time lapse in the next series, this ribosome will have moved, this one will have gone off the end. So they're making individual protein subunits. So, as it stands right here, all 5 of these are going to be 5 proteins at the end. You don't join them. They're 5 copies of a particular protein. Does that make sense? Okay. Sometimes I went so fast, some of that might not have been clear. Yes.
When you create a polycistronic mRNA molecule within a eukaryotic mRNA, and you only have one start codon, I don't understand why it's unable to then read all of those and then create the...
So first of all, just to let you know, you don't see those polycistronics, but you could do it in the lab. So is that what you're asking when you do it in the lab? Okay, that's because, let's say there's a downstream start codon. There's multiple cistrons there. Well, how do you begin translating those? If we're in a eukaryote, you have to start from the very end, and you can only begin from the very first start codon. Okay, and then it'll go along and translate, and then it'll hit a stop codon and falls off. So it doesn't matter if there's anything down there, it'll never reach it. Does that make sense? Okay? I'll hang out up here if you guys got other questions.